Okay, I think I'll get started. Good morning, everyone. It's a pleasure to give the talk today. I've been 2 weeks now at CGSI, and it's been really fun. I gave a tutorial, I think, last Monday. Today, I'm switching gears completely to talk about a very different topic, which is more a research topic currently ongoing in my lab. The work I'll talk about is actually recently put on the bioRxiv. It's the work of my graduate student, Jay Scaring, former graduate student, I should say, who just started his postdoc. And I'll be talking a bit about the paper that's on the bioRxiv, but I'd sort of like to give a bit of background as to where it came from also as part of the talk. And this is a computational genomics summer institute, but I will be discussing a sort of partly experimental project. I think it's appropriate here because there's going to be a lot of interesting computational questions emerging from the application of the technology, I think, and that's why I decided to present it today. So the project is about multiplexing, and multiplexing is a sort of generally very good idea, and it comes up all the time in genomics. And it sort of actually goes back to Hedy Lamarr. She was a very famous scientist who came up with the idea of frequency hopping. This sort of came out in a military industrial setting where there's sort of a problem where you transmit signals to torpedoes, they can be jammed, and she had this sort of very interesting idea of splitting up a single signal into multiple different frequencies, hopping around in a sort of pseudo-random schedule, making it very hard for an intermediary to jam the signal. I think that next week there's the final Hollywood Bowl trip on Wednesday, and if you go early, I urge you to go visit right outside the Pantages Theater. You can go visit Hedy Lamarr's star. She was also a famous actress, and she has a star on the Hollywood Walk of Fame. It's my favorite one. So I urge you to go and visit it. Multiplexing also has another meaning in English language, and I'd like to sort of discuss both today. The second is that you have many elements in a sort of complex relationship. And I'm going to sort of make the point that the work I'll talk about today partly came out of sort of multiplexing my lab. And I think that's an interesting thing for the graduate students here to think about as you're starting on your PhDs or trying to end them, whatever the case may be. So I'm going to tell a bit of the origin story of the project. And to do that, I kind of have to go back to 2010. And you'll see in a second why I want to talk about the origins. I think that there's some interesting lessons to learn. So in 2010, I was a mathematics professor at UC Berkeley. The definition of a mathematics professor is somebody who teaches calculus, and so that's exactly what I was doing. I was working a little bit in computational biology and so on, and I got the privilege of becoming what's called a Miller Professor. Miller Institute is a really special kind of program that's hosted at UC Berkeley, where both postdocs, faculty, can do a sort of funded postdoc for a few years as part of the institute. Faculty can be visitors there. And it's a very special place in that it covers all of science. It's a fairly small group of people. And you meet every week for lunch. And one person gives a lunch talk about their work in a sort of accessible way. And there's scientists from many fields all listening to the talk. I think it's a wonderful thing. For those of you who are about to graduate, you might consider applying to be a Miller postdoc at UC Berkeley. It's a really wonderful program. So that particular year, I was a Miller professor for a few months, semester, and I gave a lunch talk, actually on February 23rd, 2010. And what I wanted to do is tell the Miller Fellows, they're called, what I work on. And the focus of my talk was this slide, which I'll get to in a second and tell you how I got there. So to get to that slide, I'm going to, like in fast forward motion speed, deliver my Miller lunch talk very quickly. I started with giving the whole history of DNA, like when the molecule structure was discovered, which hadn't been that long ago, how like people started sequencing DNA, about 2 decades after that, and how that had led up to the human genome. And I told a pretty good story because I had been at the MIT now, the Broad Institute, right when that was happening. So that's how my career in this area actually got started. And I pointed out that, it cost a lot of money to sequence the human genome. This is the kind of thing that's not that exciting now. 10 years ago, it was also not that exciting, but more exciting now to talk about this. Okay, so that's what I did. And then I kind of went on to talk about the fact that following the human genome sequencing, which really sort of completely, the draft was sort of done around 2003. This is 2010. You have to put yourself back in those days. There was a lot of excitement about, all these technologies to improve DNA sequencing. And, Oxford Nanopore wasn't really a thing yet. But Illumina was already a bit of a thing. And I pointed out, how sequencing was getting much cheaper very quickly. That's sort of what I wanted to say. And that was a lead-in to my final point, which is what is everybody doing with all the sequencing? So now I have the benefit of hindsight. So this slide actually comes from my former student Atif, who graduated in 2015 and made this for his thesis. So this goes beyond 2010. But I like this slide. It shows, on the x-axis the year, and the y-axis is sort of the cost of sequencing in the inverse, how many mega base pairs you get per $1,000. And what's kind of nice about this slide is a lot of information on it. The different technologies are in different colors. And the diameter of the circles are proportional to the read lengths somehow. And the thick boundary on the outside is the error rate of the technology. So you're going to sort of see here that, there's a bit of a trade-off between the length. And, you've heard a lot about that now at this workshop. We can now look back and see how, for example, Oxford Nanopore and Pacific Biosciences have been transformative for genome sequencing. That wasn't so obvious yet in 2010. Okay, and then I kind of said this in 2010, but somebody made this slide afterwards in 2015, but it makes my point for me better now, so I'm going to use it. Which is that, if you extrapolate out sequencing capacity with time, we're basically, in a couple of years, it's actually not very far, depending on how you extrapolate blue, yellow, or red, we will have sequenced everybody on the planet. I think, we're looking at, 5, 10 years from now. I think that's actually pretty realistic. It's a little bit hard to grasp today, but if you look back where we were 2010 to today, it's hard. People under-predict the long term. They over-predict the short term. But I think in 10 years, we're going to have sequenced basically most of, maybe not all humans, but we will basically be doing this in the standard way. So what's sort of the point of sequencing? What is the end of sequencing? And that's what I was talking about in 2010, was this picture, which I actually took from a paper by my now colleague Barbara Wald at Caltech. She wrote a really nice opinion piece around, I think, 2008, in which she sort of drew an analogy between DNA sequencing and Thomas Edison's discovery of the phonograph, the record player thing. Maybe you don't know, but yeah, it's popular again. We're in retro now. It's all cool to play records. And the thing is that, the interesting thing is that when Thomas Edison actually developed the record player, he had actually meant for it to be an answering machine for the telephone. But that's not sort of how it ended up. And she was sort of saying that this was a joint paper she actually wrote with Rick Myers. They, I should say, were saying that that's sort of what happened with DNA sequencing in a way, is that, all this technology development between 2000 and 2010 was all geared towards getting cheap genomes. And that's a good thing, people are doing that. But it had the side effect of being useful for doing what we now know as RNA Seek, and this Seek and that Seek and everything Seek, ChIP-seq, blah, blah, blah Seek, now single-cell RNA Seek, by virtue of doing biochemistry to convert measurements you want to make into DNA sequencing. And so I talked about that, and I had just been working on RNA Seek. And so I wanted to deliver this message to the Miller Fellows that this was an exciting thing in our field. Again, I didn't have this nice slide, which was made by another former student of mine, Akshay Tambe, but I'll show it now because I like this slide. It illustrates in this kind of transformation of going from the measurements you want to make to the sequencing providing the measurements, you have two things basically you're working with. You have this whole toolkit of biochemical reactions. You can do things like modify groups on molecules. There's all sorts of modifications you can make for molecules. And a good biochemist will have sort of this dictionary in their mind, if you like, which, if I want to change this, I could use this. If I could use this, this will do this. And it's the kind of thing where, as a mathematician, I had no idea when I started in this. There's other things you can do. You can induce mutations in DNA. You can change bases. That's sort of another operation, if you like. And we have various assays like bisulfite sequencing, Parclip, all sorts of assays that are based on that. Okay, not so much modifying groups on molecules, but actually changing the bases themselves. There's selective ligation is a kind of another toolkit. That just means fancy language for you can cut molecules, and you can cut them in ways that are specific to their sequences, for example. And there's various ways to do that. You can also just straight out cleave molecules, and there's a lot of interesting tricks for how to purify out certain molecules based on their sequences or based on various beads or other things you've attached to them. So this whole toolkit has basically provided a playground for creative biochemists to think of things you could do to DNA, which when you sequence them, you could decode. And on the sequencing side, there's sort of basically a couple of ways you can classify for how you get information out of DNA. Of course, you can just sequence DNA for the DNA sake, but here we're really using the DNA sequencing to extract information that we encoded. It's a different thing. And you can count events with barcodes, namely just sequences you synthesized into the DNA. You can also use synthesized sequence that you put in to distinguish objects. And you can identify locations. So you can take the sequence you got and you can align it to a reference and find out where it came from. And finally, you can look at specific mismatches that you put in and find out what they were. And you can also see where your sequences ended. If you had done some kind of cleavage, then you can find out, okay, that's where that cleavage actually happened. So it's actually been very interesting for me as a more math person to see this. It's sort of like a little bit, I think, like in computer science, you have these reductions from one problem to another when you're proving things like, for example, NP completeness. And here it's the same thing. You're trying to measure something and you have these reductions you can do, these operations that you can go decode. So that's what we were playing with back in 2010. I mean, Barbara Wald was one of the pioneers. She published one of the first RNA Seek papers and ShapeSeq papers. And I was learning about this at the time. And now we know that there's literally hundreds of assays that have been developed based on the schema. Most popular, as I said, is RNA Seek. I think everybody in this room by now knows about RNA Seek, so I won't go through the assay, but it was in a sense one of the simplest implementations of this concept. You want to measure abundance of RNA molecules, but you're going to end up doing that by doing DNA sequencing. And in retrospect, it's a pretty obvious assay, but when Barbara first started doing this, I think around 2006 and 2007, it was not obvious that this would work. The sequences, the reads were very short. I feel like an old guy in the room, but I think there were, back then, 25 base pairs. Later, there were 32. It wasn't obvious this would work, but it worked. So I was giving this talk very much to a room like here, talking about this general new idea. And in the room was a young Miller Fellow who was Miller Fellow from 2008 to 2011. His name is Julius Lux. He is now a professor of chemical and biological engineering at Northwestern. And he listened to my talk, and he was working on on other things. He was interested in structure of molecules. And he thought, oh, this is kind of interesting. Maybe we could develop one of these assays for my problem. I would have never had the courage to do that kind of thing. But Julius was doing, practicing a habit which I encourage all of the graduate students here to practice. I learned this from Feynman. One of the possible good things he actually had to say was that, it's really good to come into every talk with like sort of 3 or 4 things on your mind, always the same things, and you extract from the talk for yourself. So that's what Julius was doing. So he proposed that we develop an assay called ShapeSeq. And this assay kind of had the same schema. You know, you want to measure, in this case, the structure of an RNA molecule. And his idea was that we would do some interesting biochemistry and extract out something about the structure. And the assay is actually kind of interesting. There was already a technology called SHAPE. Now, SHAPE stands for selective 2' acetylation, 2' hydroxyl followed by paired end sequencing. So it's blah, blah, blah. Long acronym, but it's actually conceptually quite straightforward. What you do is you take your RNA molecules and you modify them in a way that when you reverse transcribe the RNA, if a modification happened at a site, the reverse transcriptase will drop off there. So you'll end up with a DNA molecule, not RNA, but DNA, that is like ended where you had made this modification. Now, why is this useful? Well, this modification turns out to preferentially happen on nucleosides or sites on the RNA that are unpaired. So, you know, RNA has a structure. It has a three-dimensional structure, but you can also represent two-dimensionally just what's paired to what. And so by doing this assay, you might learn something about not the actual 3D structure, but which bases were paired to other ones, because they will get less modified and you'll have fewer reads that ended there. That's kind of the concept that Julius had in mind. And we actually did it. So it took about a year of hard work. So it was actually an interesting collaboration. Aviran is my former postdoc. She'll be talking here on Tuesday, actually. I'll talk about her work in a second. And she developed all the math that's necessary to actually make this assay happen. You can't just sort of naively do this, for a reason I'll show in a second, because there are a lot of biases that come out in the sort of experiment. And Julius, working with the Adam Arkin and Jennifer Dowd in the labs, sort of developed the biochemistry, and Illumina actually helped us do the experiment. So, yeah, the experiment is pretty straightforward, kind of like RNA Seq, except now you do these modifications on your molecules. You'll also do a control sample, and you can kind of see this on the top left. You get these little red dots there where you actually made a modification. And then you're going to go and sequence and find out where they were. And so you'll get data, which is just reads. You already know the RNA sequence, so you align them and you find out, okay, this read ended here, this one ended here. Okay, I had a modification there, I had a modification here. So where Sharon came in, the hard part here is that if you have two modifications on a molecule, you'll drop off at the first and you'll never measure the second. So there's this kind of interesting hidden aspect to some of the data. And I won't go through the math today because that's not what my talk is about. But Sharon had to develop a model, which she did. And it was sort of an interesting model, and you had to do inference just to get out the measurement. And following that, of course, you now have this new modality of data that you can then do really interesting computational biology on. You can ask, can you better fold molecules three-dimensionally with this kind of data, and all those sort of questions. So I got very excited by this, and we got some nice results in this paper, and I thought to myself, well, I kind of want to do more of the same. And I never really thought to have a lab, because that would be a really crazy idea for a person like me. And now I've actually, after a couple of years, done the experiment of developing a lab, and I can confirm it's a crazy idea. So how did I get started? So as part of being a Miller professor, they gave you a little bonus for like research funds to go and do something fun. So I got $5,000. So I actually paid for my own first experiment. It's quite a liberating thing to do. Because it turns out if you pay for an experiment, it's your data. So that's sort of a good thing, because then you can be last author and all that sort of stuff. So I tried to do that. I actually went to talk to a dean at Berkeley, and Mark Schlissel very kindly helped me get a little small amount of wet lab space. And my initial idea was sort of to launch a little wet lab where we could do the kind of thing I'd done with Julius, right? So I wasn't that, all about like actually doing real experiments where I learned something about biology, but more like do biotechnology so that I could learn about biology in new ways. So that was kind of the idea. And I started recruiting students, and I bootstrapped up a lab, and I can tell you more about that some other time. I won't say too much today. My first student was very brave, Shannon Hately. She was a graduate student in molecular biology who agreed to set up my lab. We sort of decided not to just hire some professional to do it, but to do it ourselves, because that was the whole idea, is to learn how this works. So I've had 4 wet lab students so far, and the work I'll talk about today is of Jace, who, as I said, just graduated, and I'll be talking about his thesis work. So his work is in the single-cell genomic sphere, and well, why is it good to do single-cell genomics? I like very much this slide. It's from a paper that I had nothing to do with. It's a nice review written by Cole Trapnell, also a former student who's now at the University of Washington. And one of the things Cole does is think about biology quite deeply, about sort of what's the goal of what you're trying to do. And not just do, but think about what's the goal. And I like this slide because it sort of goes through the thought experiment of, like, why would you actually do single cell experiments other than the fact that you can? And basically the rationale that Cole posits here is that Simpson's paradox in statistics can harm you when you're averaging over data. And so what that means is, I think this top left panel is a good one. If you're comparing, the expression of 2 genes, you might find that they have a negative correlation. But when you break it down by their type, you might find positive correlation. So, basically averaging can be misleading in sign. I mean, in a very substantial way. You might believe the totally wrong direction of what's going on. And all of these different examples here, what's now called pseudotime, have to do with this effect that when you break down an average into the constituent pieces, the phenomenon you're studying might have a reversed effect. So I think, this idea that single cell will be important to resolve questions having to do with the nature of this paradox, I think is borne out to be true. Single cell isn't always the right tool, but in some cases you learn because you identify exactly situations like this. So where are we today with respect to the technology? I just have to talk about this before I talk about our work, so we have some context. Now, this is kind of a hard topic to talk about because the technology changes every week. And no matter what I say, I'll offend somebody who just got a new technology paper published on, I'll just check Twitter while I'm giving this talk. All right? Also, the technologies, which there are many now, have many properties that you can compare them by. And it's sort of hard to settle on one as like the canonical example. So the sort of chart I'm showing here is kind of data based on droplet-based technologies. And the work I'll talk about today is sort of, based, I should say we use this kind of technology to do the work, which is why I'm mentioning it. But there are many, many technologies. So in our own lab, we have a rig called the DropSeq rig, which is like a particular kind of method that was published a few years ago. It's sort of open source, do it yourself. It's a microfluidics-based, droplet-based single cell technology. There's a company called 10x Technologies that sells a machine to do this, also a company called Bio-Rad to do the similar thing. Order of magnitude today in a sort of typical experiment, you could get maybe about 10,000 cells. You might measure about 1,000 genes per cell with these kinds of microfluidics technologies. There's also one called InDrop. And you will pay about $3,000 or so. And you can trade off. If you'd like more cells, you can do that, pay more money. You can actually sequence fewer cells, but get better information about each gene. So, there's a lot of nuance here. But to first order, you should keep that in mind. Just to give an example, this is from one of the papers by the 10x company, where they published a sort of showcasing last year, doing single cell in blood. And the sort of point of this paper is that, they clustered the cells, and you can see that you get out clusters which correspond to cell types in blood. I mean, that's just sort of ab initio not obvious that this would happen when you do a single cell experiment. And what's kind of nice about this paper is that separately from just the naive single cell from peripheral blood, they actually purified cell types in blood by antibodies that are specific to markers for specific cell types, and sort of aligned the data to show that that this really is these cells really are monocytes, because you get the same cells when you purify out monocytes by looking at CD14. So I think that it's fair to say that this kind of works, and it's actually a little bit magical that you can actually do this. There's a lot of work in this area, and it would make for an interesting summer program just to talk about the methods going right now into just this one single technology. This is a paper I like that was just published, I think, about a month ago. It is the zebrafish, period, like the zebrafish. So there's all the cells in a zebrafish, during development. So you can actually, they're painting here the cells by the different cell type. And they're painting here the cells as they develop in time. So you can see they start out here and they're developing. I mean, so it doesn't look like a zebrafish, but it is the zebrafish. And actually, yeah, with these kind of pictures they made with this thing, TSNE, you could probably make it also look like an actual zebrafish. That's a good exercise for those of you who are staying on for the program, is to figure out how to get a TSNE plot to look like a specific shape. All right. But anyway, I think this is totally amazing. Like in one experiment, basically, you learn an animal. I think we can expect this to happen now for many, many, many different animals. We're going to learn how all the cells are generated from single cell during development, and what the differences are between these cells. Pretty remarkable stuff. Okay, so back to multiplexing. So what were we working on? So Jace, started, actually, he was working on a collaboration, and one thing led to another. So I won't tell the whole story of how we got here, but that's interesting in and of itself. But eventually he started working on a technology to multiplex single cell experiments. Now, what do we mean by that? So let's say you have many different samples that you want to do single cell RNA Seq on. So right now, you got to take one of those samples, and like I say, you pay about $3,000 to get about however many cells your technology will actually produce for you. But what if you want to do many, many, many different samples? Well, right now, you got to go out and pay $3,000 a pop, and you'll get quite a lot of cells, and if you wait a year, you'll get even more cells for that sample. But you don't have a knob you can tune, which is to say, I'd like to do more samples and get less cells from each sample. It sounds like it's a technical matter, but it's actually, I'll try to argue, really fundamental trade-off you'd like to make. And how would you make this trade-off? So the way we went about doing this is to put a tag inside the cells of one sample, so that the first sample there is tagged with one DNA molecule that we order, so we know what its actual sequence is. The second sample is tagged with another molecule. We're literally going to put these tags inside the cells, so that when we go out and do the sequencing, the cells will all be mushed together and sequenced, not just sequenced, but assayed at once. And you wouldn't know which cell came from which sample, except that the tags inside your samples are going to tell you where they came from. I'll show you how that works. This will allow you to multiplex your samples. For example, in this experiment, in order to get all of this information, they had to pool lots of zebrafish embryos. But one thing you might be really interested in, and would be hard to do in other animals, you can maybe do it in zebrafish and get this information in other ways by using zebrafish with different genetic backgrounds. But in the general case, you might want to know not just how the overall development works, but you might want to break it down by individual. Right? So now we're going to be able to do that sort of thing. So how does this work? It's really actually not that complicated, and it's kind of interesting, so I'll go through it quickly. So first we're going to barcode the cells. We're literally going to put DNA oligos in them. We call them tags. the way that the single cell works in these microfluidic assays, they're all a little bit different, but this is sort of an example from 10x, is that now our cells are going to go through these channels together with beads. The cells now have these special colors, if you like, because we've tagged them. And then right here, you'll have a little junction where you'll basically create an emulsion where you now have a little aqueous droplet, where inside you'll have your cell and you'll also have your bead. Your bead has also been synthetically made to have its own DNA barcodes, so that when you go out and do sequencing, you'll find out, that the reads, the sequences that came from this cell had this barcode, and from this cell had this barcode, so you'll know if they were from the same cell or different cells. But now we'll actually have, in addition to this library that tells us, oh, we'll have, RNA molecule together with tag. We'll also have, together with cell barcode, we'll also have our tag together with cell barcode. So we'll be able to match the barcode to the tag. So we'll be able to disambiguate cells by their samples. Okay, so I've already mentioned that there's lots of ways to do that. Actually, in our lab now, we're developing a sort of low-cost version of the ShapeSeq instrument, which I just thought I'd mention for those of you interested. And, we order these sequences. They have a very specific form. We get to design the cell tags. The chemistry was not entirely novel. All the ingredients come from other settings. I think Jace's work was to get this all to put together in this application and to get it to work. It took him a while. These kind of things have to be optimized. And I think I'm running way behind time, so I'll try to go a little bit faster. So what comes out of this is data like this. So this is a very simple proof of concept we did, where there are 4 samples, and each sample we've tagged with 2 different tags, okay? And then you get, 2 libraries. So, there's actual RNA, which I'm not showing here, but then we've actually sequenced the tags, and that I am showing here. So in this picture, each column is 1 cell, and these are the tags that showed up there. It's pretty clean and nice. And this is a kind of visualization of those counts information, okay? So what you would expect to see is 4 different clusters, because there are 4 different samples here, right? We tagged each with 2 tags. And if you do the math for a second, 4 choose 2 is 6, the number of ways to pick 2 out of 4. So you might accidentally sometimes actually put 2 cells from different samples together. That could happen by accident. And so that's what you get all these colors here, okay? Which are mixed. These are doublets, they're called in single-cell RNA-seq. So you measure a couple of those as well. Okay, so this basically works. And I don't have a lot of time, so I won't talk too much about all the experiments we did. This was a very interesting species mixing experiment we did, where you have, you mix human and mouse, and you tag them with 2 tags, 3 tags, 4 tags, 5 tags. We wanted to show that you can tag one sample with multiple tags to demonstrate that with a fairly small number of tags, you can pick and choose among them to generate an enormous number of combinations. So you can essentially tag very large numbers of samples, like thousands, without trouble. And it works quite nicely. And then we did a sort of proof of concept experiment, and that's what I really wanted to talk about and get to, because that's where the computation part comes in. So what we did is, this is collaboration with another lab at Caltech, Matt Thompson's lab. We did a perturbation experiment on neural stem cells. These are a kind of cell, and we perturbed these cells in multiple different ways by changing the extent of perturbation of different chemicals. You put into the stem cells, and we varied retinoic acid, scriptase, BMP4, EGF, FGF, and so we created basically this cube of experiments. It's a total of actually 96 experiments, and each one is an entirely different set of cells that have been somehow perturbed, and then we did single cell on those set of cells by multiplexing. Because we can multiplex, instead of this experiment costing about 100 times 3,000, which would be $300,000, which I can't afford, all right, not even close. We paid about 2 experiments worth, which I can't afford, and we got, just a handful, a couple of hundred cells per sample, which is actually great because a couple of hundred cells is actually quite a lot, if you think about it, to tell you about a sample. In fact, 2 or 3 years ago, that would have gotten you a paper in Nature, right? Because it started out with 20 cells, 30 cells. So that's the data we got. And what I wanted to talk to this audience about, to all of you, is that actually how to analyze this data, how to think about it, is actually conceptually challenging from a mathematical point of view. How to organize this data has challenged us because you have, in a 96, array, 96 cell array, which has in and of itself structure because you have gradients of the perturbations you've done. Inside each of those, you have an entire single cell worth of data, which is a whole matrix. All right, so how do you think about this? So, you can cluster the data, which we did, find different cell types, and now you can actually try to figure out which experiments map onto which of the types of cells here. For example, we find that there's one set of cells that are basically dead. We killed them, and you can find out which experimental conditions took us there. So you can kind of do that kind of analysis. My favorite analysis that we did from this is in this picture where, and this is the last thing I'll say, is where you look at each of the clusters that you got from the whole data set, not partitioned by sample. You look at, excuse me, how many cells were present in each of those clusters from each of the different types of experiments. And you basically PCA that data to get this picture, which is a little bit abstract. So every point here is not a cell, but it's an entire experiment. All right, and so what you have PCA'd is how much of each cluster was represented in that sample. So you can see here that, as you vary BMP4, you change the representations of the clusters in your sample in this direction or that direction. You can see when you change retinoic acid, you change the representation of the clusters. So it's a kind of visualization of the entire experiment where every dot is one whole sample. So, if I had more time than my 45 minutes, I'd talk a lot about ideas we have about how to think about all of this. But I think that this kind of technology, actually, in addition to our paper, already before us, there was a multiplexing paper from the Satija group using antibodies. There have been a couple of papers since. This will be a totally standard method now, which means that every single cell RNA-seq experiment won't be one experiment, but will have replicates and multiple conditions, thousands of samples. So you got to wrap your head around all of this, and that's the challenge for you guys. I think I'm way, way out of time, so I'll stop here and thank, again, CGSI for a wonderful 2 weeks, and thank all of you. Yes? Yes. Okay, so yeah, I went through this very quickly. Let me slow down a little bit. So first of all, when you have, in this kind of approach to single cell RNA-seq, you have these droplets. Inside every droplet, you have a bead and a cell. So basically, the way you do single cell this way is that every bead has DNA oligos attached to it, so that every bead has the same DNA attached to it. And another bead will have different DNA attached to it. This DNA also has a poly A tail to capture poly T tail, to capture polyadenylated transcripts. So the poly T's at the end capture the RNA, and then you make cDNA. And the way you now know the cell is you get paired end reads out of this whole experiment. One end is just RNA from transcriptome read, and the other read has this barcode. So every single RNA molecule in one cell will have the same barcode, modulo errors. And then you can group all those together and know they came from one cell. These were the reads there. Because the bead had the DNA oligo together with the poly-T to capture the polyadenylated transcripts. One consequence of this is you only get 3' reads. You get 3' sequences. But that's how the single cell works. Now what we are doing is we are in addition putting into the cell our own DNA oligo that is sort of a fake transcript. It looks polyadenylated. It looks like a polyadenylated mRNA. And so it also gets sequenced. No, it will be, it's totally separate actually. But it will now have this and the barcode. But that same barcode was there attached to RNA, so now you can kind of transitively group the reads with the tags. And it has actually a lot of, so actually it's done in the sequencing 2 separate libraries, in fact. And it's really powerful. I mean, I don't have time to go into this today, but for example, one of the challenges in single-cell RNA-seq is to decide whether a cell is real or not, whether the data from a cell is real or not. And now you have this totally orthogonal data to help you do that. Yeah, it's sort of, oh, it saturates, yeah, it's totally saturating all the cells. Yeah, it's saturating all the cells, and we can sort of see that. So, yeah. Yes?
You mentioned you're trying to get the control number of samples.
Yes.
What about the number of samples per cell?
Okay, that's a very good question. And one we've looked at a bit. We have one of the attendees, I don't think he's in the room right now, but it's CGSI's Vasilis Dranos, my postdoc, and not working with me, but with David Shea, they have a paper, asking exactly that question from a theoretical point of view. And they have like actual guidelines for what would be optimal by some criteria. So that's a very good question. It's kind of a complicated discussion to have because there's really 2 different things. There's how many of the transcripts in the cell are you actually having access to and probing? And it's not the majority of them. It's a small minority, let's say with 10x, maybe only 10%. And then there's how many reads you're expending in your sequencing to measure those transcripts. And those are related but slightly different things. So yeah, we have thought about that. And a lot of what we're doing is sort of simulating different scenarios. How many cells do we need? How many transcripts per cell? So forth and so on. But I think generally good intuition is it's better to have more samples than fewer, better to have more cells than fewer. And I would happily trade off samples for cells and cells for transcripts, generally speaking. Yeah. Yeah, so you can imagine a lot of different applications, right? So, for example, most straightforward application is, right now in single-cell RNA-seq, there are very few experiments that have a replicate, which for anyone who's taken the basic stats in your program here should kind of be worrisome, because you get a one-shot, like these are the cells, this is ground truth. You have no idea about the variance in your experiment. And the reason that people haven't been doing replicates is not that they don't know statistics, but because it's very expensive to do it. So, by multiplexing now, it makes it much easier for you to actually make replicates for yourself in your experiments. Secondly, you can do kinds of experiments that would be totally intractable otherwise. Like, we're pondering various experiments where we're in the course of multiplexing hundreds, I think soon thousands of different samples. Let's say from many, many different patients, if you like, in some setting, or from many, many different individuals. If you're studying the zebrafish, let's say, you might want to look at lots of zebrafish that were raised in different conditions or have different genetic background. You can imagine a lot of cases where being able to do, hundreds of samples would be very liberating. For example, this experiment that we did here in this paper has 96 samples. It's around one of the largest single-cell RNA-seq experiments in terms of number of samples done to date, because to do this without multiplexing is a hundreds of thousands of dollars experiment. So, so maybe very big places with lots of funds could do it. Now any lab can do it. You're compromised on the number of cells per sample. But it depends on your question, right? Like if your question is to go in peripheral blood and find the really rare cell that's lurking there, that's a biomarker for your disease, okay, this isn't the appropriate thing to do because you want to get lots and lots of cells. But, you know, if your experiment is really based on just measuring what's the relative composition in some known clusters of cell types in your sample, yeah, then this is fine. People have written a lot of high-profile papers already, and I don't mean in good journals, but I mean just with really interesting results. In the last 3 years of single-cell RNA-seq, where the majority of papers have been published with less than, you know, a few hundred cells, right? It's only in the last year or so that you start going up to 10,000. So there's plenty you can say by just measuring, say, 1,000 cells instead of 10,000. Okay, that's a very interesting question. So, yes, there's variability. It can be a feature, not a bug. So, for example, this is sort of a good example of this. So, human and mouse cells have different sizes, and we're able to tag both here. Now, that's not to say there aren't limits on what we can tag. Yeah, there are going to be types of cells we probably can't tag for various technical reasons. Although this tagging approach is not antibody-based, so it's, you know, blind to the type of cell, really. Like you can tag any animal. But what you see here, actually, and we have other data in our preprint, but you can see it a little bit here. You know, this cluster over here is a mixture of human and mouse cells. And if you look, you see there's a gradient from yellow to green. And we believe that this is, and we've looked into this a little bit, that basically we're measuring more tags in the human cells because they're much bigger. And so actually our assay is maybe sort of providing an indirect measurement of cell size, which we're actually looking at because it's quite interesting. So the answer is yes, there can be limitations. You know, there's all sorts. One limitation that one should disclose up front that's very important is that what we currently publish is working on fixed cells. Methanol fixation is how we produced all this data, and that actually changes the transcriptome, and it's not like, we can't do this right now very well with live cells. We've done some experiments, but it doesn't work as well. We think that's eminently very fixable and will be done either by us or somebody else shortly, but right now we're working with fixed cells. So, you know, so there are various caveats, but you know, they'll all be ironed out, I think, pretty soon, and this should be pretty standard and straightforward to do. Yeah, the antibody tagging is working, for example, with live cells. So, yeah. Great, thank you very much.